Hello everyone, welcome to the NN Classroom. Today I'd like to share with you a multiple choice question number 26, extracted from the Specimen paper 2016, asking for a really common concept extracted in the NOS area, Nature of Science, from experimental history in two really important scientists, Hershey Chase experiment that conclude that the DNA was the genetic materials. Of course, now we know DNA is the genetic materials or the inheritable factor. But at that time, they are having a doubt whether DNA or protein should be the genetic materials. And so they conduct an experiment using bacteriophage that one side or one batch radioactively labeled the phosphorus and one side or one batch that radioactively labeled the sulfur. And each side infect the E. coli bacteria. And at the end, realize that the radioactively labeled phosphorus is the one that get into the bacteria cell and generate a signal, but not the sulfur batch. So that's why they conclude DNA was the genetic materials, right? Why? Because we should have learned that phosphorus is the distinct element that represents DNA, right? Because phosphorus only present in DNA in this case, but not protein, while sulfur is the distinct element for protein, but not DNA. If students realize the structure of these two biomolecules from topic 2, they should realize why they are choosing phosphorus and sulfur, but not carbon, hydrogen, oxygen, or nitrogen, because all these elements are not distinct in each of the biomolecules, right? But existing for both, right? So that's why those are not good choice, right? But here, remember, the idea about this experiment is that the success of locating the right biomolecule that is representing genetic materials is that we locate the one that gets into the bacteria cell, but not the one that cannot infect the cell. Cannot means you cannot be infecting, so you cannot pass the materials to the cell. Remember, at that time, these two gentlemen are not looking for the thing that remain outside the bacteria, but the thing that get into the bacteria. So that's why for this perspective, for A and B, the phages containing the radioactively labeled element that unable to infect is not the main focus, because the thing that unable to get in is not being recognized in the cell palette. So that's why answer not A and not B, because of the word unable, right? Because we are finding the thing that able to infect and so pass it into the bacteria, or you can say into the palette. Here the palette representing the cells, right? What are the cells? Here obviously the bacterial cells, right? So that's why here is concerning where the signal of radioactively labeled phosphorus is get into, or radioactively labeled sulfur is get into. Obviously we know DNA is the one that get into, right? From the infectious cycle of a bacteriophage, right? So that's, as we understand that, DNA is represented by phosphorus, right? So that's why phosphorus should be the one that infected and get into the bacterial cell of E. coli. So here means will be found in the cell palette of the E. coli batch. So that's why the answer goes to C.